We are going to speak about the CSF and the ventricles and the meninges in this video. And we will finish also with some slides about the blood-brain barrier. So starting with the CSF and ventricles, I will have a video that can help understand some of these points.
Let's take a look at the ventricles inside of the sheep brain. The ventricles are cavities within the brain that are interconnected. They're lined with ciliated ependymal cells, which circulate cerebrospinal fluid. Right underneath the corpus callosum, we find the lateral ventricle. There are two lateral ventricles, one on the right and one on the left. The lateral ventricles are separated at midline by a membrane called the septum pellucidum. This region is the third ventricle. You'll notice some pink feathery tissue. This is called the choroid plexus. The choroid plexus is a vascular tissue covered with ependymal cells that produces cerebrospinal fluid. The choroid plexus is found in all of the ventricles. The fourth ventricle is just anterior to the cerebellum. The medulla and pons form the ventral wall of the fourth ventricle. We can see where the fourth ventricle is leading into the cerebral aqueduct. The cerebral aqueduct is contained within the midbrain. The cerebral aqueduct connects the third ventricle to the fourth ventricle. And we can also see how the fourth ventricle continues on as the central canal in the spinal cord.
Okay, so we have, these are the names of the ventricles. So you see lateral ventricle, third ventricle, fourth ventricle. So there are 4 ventricles, 2 lateral ventricles, then a third, and then a fourth. So your goal here is to identify their anatomy. In a coronal frontal section like that, you see the lateral ventricles, which are in the cerebral hemispheres, then the third ventricle, and then the fourth ventricle already at the level of the brainstem. The ventricles continue into the spinal cord in what is called the central canal. In addition to knowing the names of the ventricles, you need to know the communication that the ventricles have with one another. So each lateral ventricle communicates with the third ventricle through something called the interventricular foramen. The third ventricle communicates with the fourth through a structure called the cerebral aqueduct. And the fourth communicates with the central canal. So in a different view, you have the big lateral ventricles, which can be described further as far as their anatomy, describing the tips as horns. So you have the anterior horn, a posterior horn, and then an inferior horn that projects laterally into the temporal cortex, so the temporal lobe. And then you see the interventricular foramen that allows each of them, there are 2 interventricular foramen, allows each one of them to communicate with the third ventricle, and at this point we're roughly at around the thalamus. And then we have the cerebral aqueduct that communicates the third with the fourth ventricle, which takes place essentially in the brainstem. Okay, and then a continuation, the central canal. If we put like a substance that just makes a mold of the ventricular system, it would look something like this. Okay, it's like a plastic cast of the ventricles. So the ventricles consist part of a system where the cerebral spinal fluid will circulate. Let's see how this circulation occurs. So the cerebral spinal fluid is produced by the ependymal cells. The ependymal cells are in the choroid plexus. So the choroid plexus is marked here by these red structures inside each ventricle. So the cerebral spinal fluid circulates inside of the brain and spinal cord through the ventricular system and the central canal. But it has passages to move into the subarachnoid space. So the space that is below the arachnoid. So in between the arachnoid and the pia mater. It will circulate and serves essentially as a cushion for the central nervous system, and it will be absorbed by a structure called arachnoid granulations. From the arachnoid granulations, the cerebral spinal fluid will be absorbed into the blood, more specifically into the dural venous sinuses. Okay, so we have this system. There are a couple of generalizations and simplifications we are making for the sake of simplicity, in the sense that, for example, not all cerebral spinal fluid is produced by ependymal cells. You can see the cerebral spinal fluid as having more functions other than cushioning. It can also provide provides other sources, other functions as well. But for us, we can just keep this big picture. Okay. So moving now into the meninges, we are going to speak about the meninges the same way we spoke about the meninges in the vertebral canal. So we have a dura, the arachnoid, and the pia mater. The dura, the outermost, it's the hard one, it's the most superficial. Then the arachnoid in between, and the pia, which is attached to the actual cortex. So going through this more slowly, here you have the pia. So you have here the dura, the arachnoid, and the pia. The pia is attached to the surface of the brain. This is the innermost, and the outermost is the dura. The dura may consist in some areas more clearly than others of two layers. One that is called the periosteal cranial dura, just the one that is more closer to the bone, and the meningeal cranial dura. So these areas form a space where the venous sinuses will be. And so those are the dural sinuses. So you see areas in which the dura is just the two layers are merged, and areas in which the two layers are split that form the dural sinuses. The dura has not only lines the brain from the outer part, but it also forms compartments by folding. The folds will allow then the formation of compartments which are fairly rigid. So meaning that if one hemisphere is expanding because there's a problem and it's swelling, it will be compressed against the dura. So let's just spend some time just knowing the names of those folds of the dura. So you have one longitudinal fold, it's called the falx cerebri. There will be an equivalent to separate the cerebellum. It's a falx cerebelli. And then there are these two layers that one covers, forms a tent, forms a ceiling of the cerebellum, which is called the tentorium cerebelli. And you have also a folding of the dura that is around the pituitary, which is called the diaphragm cili or cili. So that's just for you to go over them. And you have also a folding around the pituitary, which is the diaphragma cili. And this would be you just need to know the names and understand the concept that they form compartments within the cranial activity. They form compartments within the cranium. So we have the outermost, the innermost. In between we have the arachnoid. The arachnoid has the arachnoid granulations. And with these three layers, we'll be able to form spaces. We'll be able to form a space above the dura, below the dura, and a space below the subarachnoid. The space above the dura, below the dura, in this image I cannot show you the space above the dura, so let's just cover that below the dura. It's called subdural space. And the space below the arachnoid is called subarachnoid space. To show you what the space above the dura could look like, we could use this image, but let me just show you this image. So you see the dura close to the bone there. So if there is, we call this is a potential space. So if there is accumulation of blood, the space will be noticeable. That space is called extradural space or epidural space. So a space above the dura, or in other words, between the dura mater and the periosteum. So these spaces are important for us, and I wanted to give you a few slides for you to understand what the epidural space is, what the subdural space is, and the subarachnoid space. The subarachnoid is where the cerebrospinal fluid circulates. The subdural is a true space. So if you look at here, you see that there's an actual space that is between the dura and the arachnoid. Likewise, there is an actual space between the arachnoid and the pia. But there is no actual space between the dura and the periosteum. This is what we call a potential space. That will show up, will be will be visible if there is, for example, accumulation of blood. So let me show you what a hematoma, accumulation of blood, looks like in these two spaces. See, a subdural hematoma. So there's blood below the dura and the arachnoid. See that you see these white parts in the middle? That marks the falx. So there's a potential for a compression of the hemisphere. And then the extradural hematoma, which normally we need to have something stronger. The blood needs to come with a high pressure. So this is normally, we are talking about meningeal arteries that release blood into with a certain pressure that basically will open up this space, which until then is just virtual, is just potential, the space between the periosteum and the dura. So this is what an extradural hematoma looks like. Just clinical notes for you to spark your interest about these topics, but they are not going to be testable for your exam. The blood-brain barrier. The blood-brain barrier is a barrier between the blood and the brain. And I wanted to speak a little bit about it, just from the point of view of anatomy. So the diagram there shows the blood, it shows the brain, and it shows the endothelial cells in green, kind of bluish, the endothelial cells, but also shows us the astrocytes, which are represented in yellow. And so the astrocytes contribute to this blood-brain barrier. So the blood-brain barrier will functionally have as a consequence that the content of the blood cannot pass freely into the brain and vice versa. And that barrier, the two main aspects to help form that barrier are the fact that the endothelial cells have very strong tight junctions. So endothelium are the simple squamous epithelium that lines the blood vessels, the inside of the blood vessel, the lumen, and the feed processes of the astrocytes. So you see here a representation of what the blood-brain barrier looks like. Note that there is a blood-brain barrier in certain areas of the brain. Normally, we justify this, we explain this, by saying that these areas are areas that are very important to control aspects of the circulation, also the content of the blood for certain substances, and therefore they need to have access to the blood in an unrestrained way. So areas like that are the hypothalamus, for example, the pineal gland, the choroid plexus, and among other ones. And the pituitary gland, since it's written there, let me tell you also the pituitary gland.